Hello everyone. Today I want to show you how to use free software to design an inhibitor of the SARS-CoV-2, which causes COVID-19. I hope this tutorial can help you use free software to design your own drug candidates for COVID-19 and other diseases. Both SARS-CoV and SARS-CoV-2 use an essential enzyme called Mpro for their replication and infection. In the X-ray crystallographic structure of the Mpro of the SARS-CoV, a peptide substrate binds to the active site of the Mpro. So if we can design molecules that mimic the natural substrate of the Mpro, but are resistant to proteolysis, maybe those molecules will block the Mpro of SARS-CoV and the Mpro of the highly homologous SARS-CoV-2. In this tutorial, we first use a free software called UCSF Chimera to modify the structure of the linear substrate and cyclize it. By cyclizing the substrate, we aim to lock the peptide substrate into its bond conformation to the Mpro and increase its stability towards proteolysis. We expect the cyclic substrate will serve as an inhibitor to block the activity of the Mpro and thus block coronavirus replication and infection. We then perform geometry optimization in UCSF Chimera to clean up the structure of the cyclic peptide inhibitor that we just built. Finally, we use another free software called AutoDock Vina to perform molecular docking and study the interaction of our newly designed cyclic peptide inhibitor with SARS-CoV-2 Mpro. The molecular docking result indicate that our cyclic peptide inhibitor fits into the binding pocket of the protein active site of the SARS-CoV-2 Mpro. These molecular modeling studies have helped our research laboratory decide to synthesize and experimentally evaluate this cyclic peptide inhibitor. We have published the biological evaluation results as a preprint and proved that this cyclic peptide inhibitor is able to inhibit the SARS-CoV-2 Mpro activities, resist the cleavage by Mpro, and non-toxic towards human kidney cells. This current molecular modeling tutorial has also been published as a preprint, which contains an illustrated step-by-step protocol of the Mpro inhibitor design process. Through this tutorial, you will learn how to delete, add, rotate, and mutate atoms, bonds, and amino acid residues in a protein, how to perform geometry optimization, and how to perform molecular docking using free software packages, which will help you design your own drug candidates. Our structure-based drug design tutorial will start from the X-ray crystallography structure of SARS-CoV Mpro. This structure has a protein data bank ID of 5B60, which has been published back in 2016. We simplified this crystal structure so it only contains the parts that are necessary for our inhibitor design, which includes the Mpro and its natural substrate. The simplified SARS-CoV crystal structure can be downloaded from the Chem Archive website, where we just posted our tutorial as a preprint. In this website, you can find the main text of our preprint, which introduces the rationale of our molecular design, and the supporting information of our preprint, which has the illustrated step-by-step protocol. You can also download the simplified SARS-CoV Mpro structure from this website. So let's begin from opening the UCSF Chimera software. So you want to download this software from their website. Now I'm just opening it. This is the user interface of the UCSF Chimera. We always want to start from opening protein crystal structure file. So the protein data bank file, PDB file. To do so, you have to download the protein data bank file. Since I've already downloaded it, I will just open it. Go to the menu bar, use File, and click Open. find where you download your PDB file. Select it and click open. We can see now the SARS coronavirus Mpro crystal structure is here. The one shows in ribbon, that's a Mpro, and the one shows in those sticks, that's the substrate I mentioned. You can also use your mouse to change the view. You can use your left mouse button to rotate the view. You can use your middle mouse button to translate the view across the screen, so move up and down. And you can use your right mouse button to zoom in and out. Play with the different mouse button until you feel the view is comfortable. Now we want to visualize the SARS-CoV Mpro as a protein surface, which will help us design our cyclic peptide inhibitor. Now it's in a ribbon form. We want to change it to a surface. To do so, we have to first select the Mpro part of the structure. Use your left mouse button and the control key on the keyboard to control click on any part of this protein. So you can see I have a part of the protein selected in green. And if you press the up arrow key on the keyboard, it will change the scope of your selection. You press up key, it will select a larger scope from atom to residue to secondary structure and to the entire model. In this case, we press up arrow key twice. 1, 2. So now you can see we selected the entire protein, but not the substrate. You can see a green outline for the parts that you are selecting. To show this protein as the surface instead of this ribbon, you go to Action, Surface, Show. So now the protein has been shown as a surface. And we also want to change the color and the transparency of the surface to help our future molecular model design. So to change the transparency of the surface, go to Action, Surface, Transparency, and we can set it as 30%. You can see the transparency changed. And also we can change the color of the protein surface. Go to Action, Color, and let's use dark gray. You may notice that the ribbon is still hiding there underneath the protein surface. So you use Action, Ribbon, Hide. So now all we left is a gray 30% transparent protein surface and the substrate show as sticks. Now we want to zoom in to have a look at our substrate. So remember I said we can use left mouse button to rotate the wheel, middle mouse button to translate the wheel across the screen, and right mouse button to zoom in. So I'm rotating and I'm zooming in. And I'm moving up and down. So now we can see how this substrate, its natural substrate, interact with the active site of the Mpro. And we want to do some structural modification to this substrate. You may have noticed that the hydrogens on this substrate is currently missing. So we want to add some hydrogen back to the substrate. To add hydrogens to the substrate, we use the Add Hydrogen tool. We can find the tool through Tools, Structural Editing, Add Hydrogen. So this will open up the Add Hydrogens window. And you can see there are two models to be add hydrogens to. One of them represents the substrate, one of them represents the protein. In other words, one of them represents the sticks, one of them represents the surface. So we want to add hydrogen for now to the substrate. In this case, this 0.1 is the substrate. So we highlight the substrate only. And we leave other settings as default and click OK. Now you can see those small white sticks representing hydrogen. Those hydrogen has been added to the substrate. So we want to modify this substrate to make it a cyclic peptide inhibitor. So first, we want to delete some unnecessary residues, bonds, and atoms. First, we want to find the serine residue and delete it. The serine residue is at position 301 of this molecule. To find serine 301, there are two ways. The first way, we can hover the mouse cursor over any atom of this substrate, and we can see the name of the atom or bond. This is part of the phenylalanine 309. This is part of the phenylalanine 305. And if we move around, we will find part of the serine 301. And to select it, we just use Control key on the keyboard and the left mouse button, Control click on any part of this serine 301. And press the up arrow key once on the keyboard to expand the selection scope from a single atom to the entire serine 301 residue. So this is the first way you want to find and select serine 301. So I want to show you the second way to find and select serine 301. We want to clear the current selection. So go to Select, Clear Selection. So I'm showing you the second way to select serine 301. Go to Favorite, Sequence. And this window will show up. It's called Show Model Sequence. Again, we select the 0.1 chain A, which is the substrate, and click Show. So this gives us a sequence, and we can find position 301 here, serine 301 here. And use your left mouse button to click and drag until you select this letter S. So you have to see it's colored in green to make sure it's selected. And you can see here in the substrate, serine 301 has been selected. Once you are done, just click Quit. Either way, now you have the serine 301 selected. And to delete it, go to Action, Atom Bonds, Delete. So serine 301 has been deleted. Now we want to delete glycine 302. I'm going to use the first way to select the residue. So just use my cursor, move it to here, Control click on any part of glycine 302. Press the up arrow key on the keyboard once to select the entire molecule of the glycine 302. Actions, Atom Bond, Delete. And we want to delete valine 303. So here, Control click on any part of the valine 303. Press up arrow once to select the entire valine. Actions, Atom Bond, Delete. So now we want to delete threonine 304, but we don't delete the entire residue. We delete part of it. So we will leave the amide bond between phenylalanine 305 and threonine 304 intact and delete the rest part of the threonine 304. So the reason will be explained later. Basically, we need the leftover amide bond to help our future molecular design to form a cyclic peptide inhibitor. We zoom in, and we can select the parts we want to delete. So to select the parts we want to delete manually, we press the Shift key and Control key together on the keyboard and use left mouse button to select all the parts we want to delete together. Once it's selected, you can see a green outline. Now, except for the amide bond of the threonine 304, we've selected the rest parts of it, and we want to delete it. Go to Select. Go to Action, Atom Bonds, Delete. And there are two more atoms that we forgot to select. We can just select them manually now through Shift, Control, and left mouse button to click to select. And use Actions, Atom Bond, Delete. Now we have deleted part of the threonine 304. Now we want to delete the entire residue of lysine 310. So we rotate the wheel. And now we can find the lysine 310 and use Control left mouse button to click on any atom of the lysine 310 to select it. and press the up arrow once on the keyboard to select the entire lysine 310. And actions, atom bound, delete. And we want to delete the actual carbonyl of phenylalanine 309. So to do so, again, we use Control key, Shift key, and left mouse button, press them together. And to select the carbonyl oxygen, carbon oxygen bond, carbonyl carbon, and the carbon carbon bond. Now we select the carbonyl on phenylalanine 309 and actions, atom bound, delete. Now that we have deleted unnecessary atoms and bonds for this substrate, and if we look at the substrate, it's in a kinked conformation, which inspired us to link the left side of this substrate, the phenylalanine 309, to the right side of the substrate, threonine 304 amide, and phenylalanine 305. If we build a bond to link the left side and the right side, we can cyclize this substrate and thus form a cyclic substrate. To do so, we will first build a methyl group from phenylalanine 309, from the para position of the phenylalanine 309. So we want to select the para hydrogen using Control and left mouse button. Once the para position hydrogen has been selected on phenylalanine 309, we use a build structure tool. We go to the menu bar, Tools, Structure Editing, Build Structure. Then we open the Build Structure window. So this window has multiple types for us to make several kinds of structural modifications. These types can be navigated using the drop-down menu from here at the top of the window. The initial tab is Start Structure. We change it to Modify Structure. The window will show up as Modify Structure, the new options. So we want to build a methyl group, a tetrahedral methyl group. So we make sure we select element to be carbon, number of bonds to be 4, and the geometry to be tetrahedral. And note here, the residue name will be changed to unknown because it's no longer phenylalanine once we add a methyl. This is important and we will explain the reason later. Once we have everything set up, we just click Apply. And you can see a newly built methyl group. And the name of this residue is now called UNK, unknown. The carbon in the newly built methyl group is in gray color, and the carbons in the rest of the substrate, they are in tan color. So we want to change the color of the newly built carbon to be consistent with the rest of the substrate. We can go to the color option using the menu bar Actions, Color, and we want to find color 10, so we go to All Options. In the new window, we check Show All Colors, and we find color 10. We click on it. Now you can see the newly built carbon has been changed to 10. And we can close the window after that. Now we want to connect the carbon on the newly built carbon to the left of the molecule, to the carbonyl carbon on threonine 304 to the right of the molecule. If we directly build a bond to connect these two carbons, the methyl carbon will have 5 bonds on it. So we have to delete 1 CH bond on this methyl to make room for the newly built CC bond. To do so, we use Select, Clear Selection to unselect the currently selected methyl carbon. Then we use the Control key, Shift key, and left mouse button together to select both the hydrogen and the CH bond. And we use Action Atom Bound Delete to delete the CH bond. After deleting, this CH3 methyl group has become CH2 methylene group. To build a bond between this methylene carbon and the carbonyl carbon, if we directly connect these two carbons, the new bond will cross other atoms and bonds in this molecule. This will cause the future step, structure minimization, to be not successful. So we have to rotate some bond in this substrate to bring the carbonyl carbon close to the newly built methylene carbon. In this case, we will rotate the CN bond in glutamine 306. We use Control and left mouse button to select it. And in the currently open build structure window, we change the tab from start structure or build structure to adjust torsions. And we click on activate to make this CN bond on glutamine rotatable. There are 3 ways you can rotate this bond. First, you can enter a new angle value in this box. Or you can click on those arrows to the left or right side of the box to change the angle value. Or you can click and drag this circular dial. In this case, we will click on the left arrow next to the box to change the angle value to -109. By doing so, we are bringing closer these 2 carbons. After we are done rotating this bond, we will use deactivate function to make this bond no longer rotatable. To do so, we click on the bond name here, and in the drop-down menu, we select deactivate. Now the bond is no longer rotatable. Now we are ready to build a CC bond between the methylene carbon and the carbonyl carbon, and the new bond will not cross any atoms or bonds in this molecule. To build a bond, we use select clear selection to unselect the glutamine CN bond. Then we use Control key, Shift key, and left mouse button together to select both the methylene carbon and the carbonyl carbon. We're ready to connect these 2 atoms with a CC bond. In the currently open build structure window, we change the tab from adjust torsions to adjust bonds. This will allow us to build a bond between these 2 atoms. Because these 2 atoms, they're still pretty far, the software will not think the new bond is reasonable. It's more than 1.5 angstroms apart. So we will change from reasonable to all possible. Then we will click on add. Now we've built a new CC bond. Note the CC bond is too long. We can fix this problem with the future structure optimization step. One thing we want to do before we do structure optimization to clean up the structure is we want to rename threonine 304. Since it's no longer a threonine, there's only an amide bond left. If we still call it threonine, when the software is doing structure minimization or structure optimization, it will show an error message. To rename threonine 304, we can use the command line function in UCSF Chimera. We can go to Tools, General Control, Command Line. Now we've opened the command line, and we can type in the following command: SETATTR space R space type space UK2 space column 304.8. SETATTR means site attribute. R means residue. Type refers to the residue name. And UK2 is the new residue name, means unknown 2. 304.A means the position on the molecule. Because when we were building the methyl group, the phenylalanine 309 has been already renamed as unknown. So we want to rename the threonine 304 as unknown 2. Once we've done typing this command, we press Enter key on the keyboard. Now you can see threonine 304 has been renamed to unknown 2 304. And we are ready to do structure minimization. Since the bond length, bond angle, and the dihedral angles of the newly built cyclic peptide may not be optimal, we have to perform structure minimization, or we call structure optimization, to clean up the structure. When doing structure minimization, UCSF Chimera will add hydrogens to both substrate and the protein. You can see there's some missing hydrogens here. When adding hydrogens to the protein surface, there will be an error message, which is due to a known bug in UCSF Chimera. To avoid this error message, we will increase the van der Waals radii of all atoms in this surface a little bit. To do so, we use select clear selection to unselect the currently selected carbon atoms. And we use Control and the left mouse button to press on any part of the protein to select the protein. You can see a green outline, which means you've already selected protein surface. In the currently open command line, we type in the following command: vdw define space plus 0.05. So we are slightly increasing the van der Waals radii. Then we press the Enter key on the keyboard, and we slightly increase the van der Waals radii of the atoms. Then we use select, clear selection to unselect the protein, and use Control and left mouse button to click on any part of the cyclic substrate. And press up arrow key twice to select the entire molecule. Now we're ready to do structure minimization. To do so, we use the Minimize Structure tool in UCSF Chimera. We go to the menu bar, Tools, Structure Editing, Minimize Structure. This window will appear. And in this window, we will make sure both the protein and the substrate are selected. So both of them are highlighted. Both of them are considered in the minimization step. At the same time, we want to fix the protein and only allow the cyclic peptide to move freely during minimization. To do so, for the fixed atom, we go from none to unselected. In other words, the protein will be fixed. And here, I would like to select neither memorize or use memorized options. So each time we use our new setting to do structure minimization. Once they are selected, click on Minimize. The next window will pop out, which is the dock prep window. We leave everything as default and make sure both the protein and the cyclic peptide are selected. Then we click on OK. In the next window, it will ask us to add hydrogens to both the protein and the substrate. We leave everything as default and make sure both are selected and click OK. Now you can see missing hydrogens has been added back to the cyclic peptide. And also, hydrogen has been added to the protein, which changes the color of the protein surface a little bit. This will not affect the future results. And the newly added hydrogen are not selected. So in the UCSF Chimera window, we press the up arrow key on the keyboard once to make sure everything is selected. Then in the Assign Charges window, we leave everything as default and select both the protein and the substrate. And we click OK. It will ask us to specify the net charge of our newly built unnatural amino acids. Remember, since we add a methylene group and we build the cyclic peptide, the unknown and unknown 2, they are unnatural amino acids now. And we can see they are not charged. There's no charged groups on them. So we will set the net charge of both the unknown and unknown 2 to be 0. Once we click OK, the minimization step will start. The minimization will last from 2 to 10 minutes. Depends on your computer memory. After the structure minimization is done, we can see the bond length, bond angle of our cyclic peptide is now pretty optimal. And the newly built CC bond length is normal. In some cases, you may want to further modify your structure after structure minimization. For example, we want to mutate the glycine 307 to a serine 307. Serine is more conformationally rigid compared to glycine. So we want to make this cyclic substrate more rigid and more resistant to proteolysis. Luckily, in UCSF Chimera, we can do the point mutation with one single command. In the currently open command line, we type swap AA, that means amino acid. Serine, that's the residue we want to mutate to. Column 307.A. This means the position we want to mutate. So basically this command means we want to mutate position 307 from whatever residue to serine. Once we have this command, we press enter on the keyboard. Now you can see the glycine 307 has been mutated to serine 307, and the name has also been changed automatically. Note the side chain of the serine is not selected. So we press up arrow key once to select the entire substrate. After we do the point mutation, we want to do a second round of structure minimization. Again, we open the minimize structure tool through tools, structure editing, minimize structure. Again, we want to make sure both the protein and the substrate are considered in the minimization process. And we want to have the unselected part, which is the protein, fixed while we are doing minimization. Once it's done, we click minimize. And for the dock prep, we keep everything as default. We click OK. In the add hydrogens window, we keep everything as default. And we can see the hydrogens has been added to the newly built serine. In the UCSF Chimera window, we press the up arrow key once to select everything, including the newly added hydrogens. And in assign charges window, we keep everything as default and press OK. Again, for the unnatural amino acid we have in this molecule, they are not charged. So we set the net charge to be 0. And we click OK. The second round of minimization will start, and it will take 2 to 10 minutes, depends on your computer memory. We have finished building a cyclic peptide inhibitor and conducted geometry optimization within the binding pocket of SARS coronavirus protease. Now we want to study whether our cyclic peptide inhibitor can interact with SARS coronavirus 2, which causes COVID-19. We will do molecular docking to see whether our inhibitor will fit into the binding pocket of the active site of SARS coronavirus 2 Mpro. We will use a free software called AutoDock Vina. UCSF Chimera has AutoDock Vina as a plugin, which enables us to conveniently do the molecular docking within the UCSF Chimera user interface. AutoDock Vina requires us to use PDB files for the molecular docking. So we will save our cyclic peptide inhibitor as a PDB file. To do so, we go to the menu bar, click File, Save PDB. In the pop-up window, we will select Save Models 5B60-S.pdb 0.1. 0.1 refers to the cyclic peptide inhibitor. And we want to select Save Selected Items Only. So we are only saving the cyclic peptide inhibitor, not the SARS coronavirus protease. Besides, we also want to select Save Relative to Model 0.1, which is the cyclic peptide inhibitor. And we want to give the cyclic peptide inhibitor PDB file a name. In this case, we will just call it ligand and today's date. After that, click Save. Now the cyclic peptide inhibitor has been saved as a separate PDB file. We will close the current session using File, Close Session. To load the X-ray crystallography structure of SARS coronavirus 2 Mpro, we can use the fetch PDB function in UCSF Chimera. To do so, we go to File, Fetch by ID, and we select PDB, and we type in the PDB ID of the SARS coronavirus 2 Mpro. In this case, we use 6YB7. PDB ID 6YB7 refers to a newly published SARS coronavirus 2 Mpro structure. So we can easily load this structure using the fetch PDB function. And we click Fetch. This is a crystal structure of the SARS coronavirus 2 Mpro. We want to do some structure modification to it. you can see there are some solvents in this structure. There are some water molecules, there are some DMSO molecules. We don't need those solvents for our molecular docking. So to delete those solvents, we use Control and left mouse button to click on any part of the protein and press up arrow twice on the keyboard to select the entire protein. Then we go to Select, Invert, All Models. We inverted the selection. Now only the solvents are selected. Only the molecules of water and DMSOs are selected. Then we go to Actions, Atoms Bound, Delete. This way, we deleted the water and DMSO molecules. Now we want to save the modified SARS-CoV-2 Mpro structure as a PDB file. Go to File, Save PDB. Again, we gave this PDB file a name. In this case, I will just call it protein and today's date. Then click Save. The next step, you should download and install AutoDock Vina on your computer. Also, you want to create a new folder on your computer which will store your docking results. And please copy and paste the just saved ligand.pdb file and protein.pdb file into the docking folder. Also, find the folder where you installed AutoDock Vina and copy and paste vina.exe file into your docking folder. After you're done with all of these, go to File, Close Session to close the current section. Now we want to load both protein.pdb file and ligand.pdb file into the same interface. To open the protein.pdb file, go to File, Open, and find the just saved protein.pdb file. Similarly, to open the ligand.pdb file, click File, Open. Find the ligand.pdb file and open. Now the molecule shown in ribbon is the SARS-CoV-2 Mpro, and the molecule shows in sticks, that's our cyclic peptide inhibitor. We want to see after molecular docking whether this cyclic peptide inhibitor will bind to the active site of the SARS-CoV-2 Mpro or not. To find the active site of the SARS-CoV-2 Mpro, we want to change the preset from ribbon to sticks. To do so, Control and left mouse button to select any part of the protein. Press up arrow key twice. Go to Actions, Ribbon, Hide. Actions, Atom Bonds, Stick. Actions, Atom Bonds, Show. And we want to change the color to dark gray. Actions, Color, dark gray. Select, Clear selection. To find the active site in this SARS-CoV-2 Mpro, we want to color some residues in the active site in red so we can better locate the active site. We already know cysteine 145, methionine 165, and histidine 164 are the residues in the active site. To find cysteine 145, we can go to Favorite, Sequence, and this Show Model Sequence window will pop up. We highlight the protein only and click Show. Then it will give us the protein sequence. And we find cysteine 145. And use your left mouse button to click and drag to select it. And you can see in the UCSF Chimera interface, cysteine 145 has been selected. Then we go to Actions, Color, Red, to color it in red. After that, we use Select, Clear selection. And we want to color histidine 164. We can find histidine 164. And use your left mouse button to click and drag to select it. Go to Actions, Color Red. Select Clear Selection. Then we go back to the sequence. Find methionine 165. Use your left mouse button to click and drag until the letter M is highlighted. And in the UCSF Chimera interface, methionine 165 has been also highlighted. Actions, Color Red. Select Clear Selection. Now we have colored the 3 residues in the active site in red. We can just quit this sequence window. After highlighting the residues in the active site, we can show the protein as a van der Waals surface to better visualize the binding pockets in the active site. To do so, we use Ctrl and left mouse button to click on any part of the protein. Press up arrow key 3 times. And go to Action, Surface, Show. And we want to hide the sticks in it. We go to Actions, Item Bound, Hide. We also want to make this protein surface 30% transparent. We go to Actions, Surface, Transparency, 30%. Now we are ready to run AutoDock Vina plugin in UCSF Chimera. To do so, go to Tools, Surface Binding Analysis, AutoDock Vina. This AutoDock Vina window will pop up. For the output file, we click Browse. And find the docking folder. Give our docking result file a name. In this case, I will just call it docking and today's date. After that, press the Enter key on the keyboard. For the receptor, we select our protein file. For the ligand, we select our ligand file. Next, we want to define a region on the protein, which is called receptor search region, where AutoDock Vina will search possible areas on this protein to dock our cyclic peptide inhibitor. In this case, we will define a box, and this box will indicate the receptor search region. This box has to cover the protein active site. To define the box, we type in values for center and size. Center means X, Y, Z coordinates, and size means the size of the box in angstroms. You should play around with those values so the box will engulf all of the active site. In our case, we have already optimized the box size and location. We will use 13, 4, and 18 for the center, and 30, 30, and 30 for the size. After that, you can see a box show up. And this box is engulfing all of the active site. For the receptor options, we want to set the first 4 as true and the last 2 as false. Because in our cyclic peptide inhibitor, we do have non-standard residues. When we were deleting and adding bonds, we generated unknown and unknown 2, these 2 are non-standard residues. So we don't want to ignore them. Therefore, we set the last 2 as false. For the ligand options, we set them as true. For the advanced options, we set the value as 10, 8, and 3. For executable location, we want to tell UCSF Chimera where did we install AutoDock Vina. So find your path where you installed AutoDock Vina. Find the vina.exe file, have it here. Once all of these are done, we are ready to perform molecular docking. By clicking OK, the molecular docking will start. and it will take from 2 to 5 minutes, depends on your computer. You can see running will show at the bottom. After the molecular docking is done, a new window called View Dock pop up. This allows you to view all of the docked molecules with different energy scores. You can see there's a new green molecule appeared, which is the docked cyclic peptide inhibitor. Compared to the undocked inhibitor, the docked inhibitor binds to the binding pocket of the SARS-CoV-2 Mpro protease. And we can zoom in. The docked cyclic peptide inhibitor with the lowest energy score is halfway in the active site binding pockets. And the one with the second lowest energy score is within the binding pockets in the active site. You might have noticed that the newly built CC bond is missing. This is because when AutoDock Vina is creating the docking output, the output is missing the ring closing bond. This is a common problem, and it can be easily fixed by using a command. In the currently opened command line, we type bond number 3.2, column 304 at C column 309 at C1. Basically, this command means you want to build the missing CC bond in the second lowest energy structure of the docked molecule. Press the Enter key on the keyboard, and we can see the CC bond has been reformed. To better visualize the interaction of the cyclic peptide inhibitor with the SARS-CoV-2 Mpro protease, we want to add hydrogens back to it. Some of the hydrogens are missing on the cyclic peptide inhibitor. To add hydrogens, use Control and left mouse button to select any part of the cyclic peptide inhibitor. Press up arrow key twice on the keyboard to select the entire cyclic peptide inhibitor. To add hydrogen, go to Tools, Structure Editing, Add Hydrogen. In this menu, select the second lowest energy structure, docked ligand, number 3.2. Press OK. Now the hydrogens has been added to the cyclic peptide inhibitor. Select Clear Selection. Now you can visualize how this cyclic peptide inhibitor interact with active site of the SARS-CoV-2 Mpro protease. The blue ligand is the undocked ligand. If you don't want to see it, you can hide it. Use Control left mouse button to select it. Press up arrow key twice to select the entire undocked ligand. Go to Actions, Atom Bonds, Hide. We hope this tutorial will help you design your own protein inhibitor and test its ability to bind to the protein in silico before you go ahead to do synthesis and biological evaluation. We hope this tutorial will help you design your own drug candidates for COVID-19 and other diseases. Thank you for watching.